Hello, everybody. Thank you for joining us for our webinar on transdermal drug delivery. This is a joint webinar with Teledyne Hanson and Gattefossé. Teledyne is a global technology company based in California that invents, engineers, and manufactures dissolution and diffusion test instruments that set the standard for the entire pharmaceutical industry. Gattefossé is a leading provider of specialty ingredients and formulation solutions for the beauty and healthcare industries worldwide. As a specialist in lipid chemistry, Gattefossé offers a full range of functional lipid excipients for all dosage forms and route of administrations. I'm pleased to welcome today's speakers. So, Bruna.
Hi, everyone. Good morning, good afternoon. Oh, whatever you may be.
Bruna Luzada is European Sales and Marketing Manager at Teledyne Hanson. She is responsible for leading sales and marketing strategies, as well as supporting distributors and customers in the region. With a master in marketing, she began her journey in the pharma field 12 years ago, working with a Hanson distributor in Brazil, where she is originally from. Since then, she has moved to different countries like the UK and Portugal to help building the Teledyne Hanson brand within global pharmaceutical companies that are developing and manufacturing solid and semi-solid dosage forms.
Thank you, Cecile, for the introduction. It is very nice to be here today, and we're looking forward to this very informative presentation.
It's nice to have you.
Who's attending.
It's nice to have you with us. Élise Dauphin-Chanard is in charge of Gattefossé Saint-Priest Application Laboratory, whose missions consist in developing and characterizing prototype formulations, troubleshooting and helping customers develop their formulations, exploring new uses of Gattefossé excipients, and contributing to innovation in our product range. And this year was very fruitful for Gattefossé with three new excipients launched. Élise has a chemical engineer degree and a master in biochemistry. She has more than 10 years of experience in formulating with Gattefossé lipid excipients, especially for topical and transdermal delivery. Élise, we cannot hear you.
I'm sorry.
It's okay now.
That's the first one. Thank you, Cecile, for this nice introduction. I'm very glad to present this webinar with Bruna and Ashwin. Thanks for having me.
And Dr. Ashwin Patel is Director of Analytical Research and Business Development at Teledyne Hanson Research. Ashwin has been in pharmaceutical industry for over 25 years. He obtained PhD in chemistry from Veer Narmal South Gujarat University in India in 1997. Prior to joining Teledyne, he held various positions with increasing responsibilities in large pharmaceutical companies. He successfully led analytical laboratories to manage multiple research projects and team of talented scientists. His expertise and knowledge in analytical chemistry helped many pharmaceutical companies to file ANDA and other regulatory submissions to obtain on-time approval from FDA.
Hello, everybody, and thank you for your kind introduction. I'm very glad to be here. Thank you.
So thanks to our high-level speakers. Be sure this webinar will be very informative and instructive. And thanks also to all our attendees. And you are around 150 people live attending this webinar, and it's a pleasure to be there with you today. So, time to start and jump into the subject. Elise, can you remind us the skin structure and how its different layers contribute to barrier performance?
For sure. So since skin acts as an effective barrier against the environment, skin delivery remains the most difficult pathway for drug absorptions. Sorry, I have some issue with, okay. So skin forms the largest organ of the body, accounting for about 15% of the person weight. So even if the structure of the human is quite complex, it can be divided into three main layers that are connected. The epidermis is the uppermost layer on the skin, and it can be divided into non-viable epidermis, corresponding to the stratum corneum that provides the most of the skin barrier's properties. The brick and mortar model is applied to describe its structure. Corneocytes are the bricks embedded in an intercellular lipid matrix composed of mainly fatty acids, ceramides, cholesterol, and cholesterol sulfate, and is organized in bilayer arrays. Then we have viable epidermis that serve to continuously renew the stratum corneum. Below the thin epidermis lies the most robust dermis, which is very supple. Epidermis, which is a subcutaneous tissue, and it consists of the layer of fat and connective tissue that houses larger bone vessels and nerves. So there is two different options for drug substances to permeate the skin, through the stratum corneum or the appendages. So sweat glands and hair follicles act as a shut, meaning easy pathway for diffusion through the stratum corneum. This way is used for water-soluble substance or for very high molecular weight substances. The Trans-Epidemic Pathway is only accessible to hydrophilic molecules that permeate across the intact, unbroken stratum corneum. And this pathway contains two micropathways. The first, the more direct one, is a transcellular pathway where the drug has to cross the skin by directly passing through both the lipid structure of the stratum corneum and the cytoplasm of the del keratinocyte. And secondly, the more common route is the intercellular route, where the drug overcomes the stratum corneum by passing between the corneocytes. So circumventing this barrier is a prerequisite for an efficient transdermal administration of drugs. Therefore, various strategies are developed to overcome the impermeability of the stratum corneum. Effective therapy depends on both the active drugs and the properties of the delivery system. But what are the best attributes for the drug to cross the skin barriers? So in case of normal skin, a drug with a molecular weight greater than 500 Daltons drastically reduces the diffusion of the drugs through the skin. Drugs with a low melting point dissolve easily in the stratum corneum and have superior skin permeability. By its hydrolipidic nature, the stratum corneum allows the absorption of amphiphilic and relative lipophilic molecules, which are log P between 1 and 4. Drug penetrates the skin better in non-ionized form. The ionization of the molecules will drastically decrease the absorption. It is therefore necessary to know the pKa of the drug. And finally, the molecule should have a good potency, meaning that the dose should not be below 20 milligrams a day. So there have been a concerted effort to investigate and develop novel strategy of maximizing the amount of permeant crossing this barrier. Innovative approaches focus on altering the drug vehicle's interaction to enhance the partitioning into the stratum corneum or modifying its structure to make it less resistant to drug diffusion. Alternatively, energy-driving methods have been employed to propel drugs across the skin. So starting with vehicle-drug interaction. Subsaturation. This method consists of increasing the concentration of the dissolved drug which result in an increased drug flux with increased thermodynamic activity of drug. Vehicles and particles with SLN, which are submicron colloidal carrier, primarily composed of fatty acid or mono, di, or triglyceride. So SLN with an enriched cells facilitate the rapid and burst release, whereas particles with a drug-loaded core lead to a sustained release. Now, optimizing skin delivery by bypassing removing the stratum corneum. Microneedles, for example, are small micron-sized needles which, when applied on the skin, reaches the stratum corneum to create microcanals through the skin, thereby assisting drug delivery. Energy-driving methods with ionatophoresis, which is a century-old technique whereby an electrical potential gradient is used to drive solute molecules across the skin. Finally, the stratum corneum modification with hydratation and penetration enhancer. These agents reversibly reduce the barrier properties, thus the resistance of the stratum corneum to drug penetration, and allow drugs to penetrate more readily into the viable skin tissue and in some cases also into the systemic circulation. So ideally,
let's have a poll question and make our audience work a little. So I will share the question. Hope you can see it. Yes. So among the strategies Elise has been talking about, which one are you using in your own laboratories? Vehicle-drug interactions, vesicles and particles, stratum corneum modification, stratum corneum bypass or removal, or energy-driven methods? Let's have time to vote. Let's still have a few seconds for everybody to have time to answer. So I will close this poll and show you the results. So vehicle-drug interactions come first, and vesicles and particles just behind, and stratum corneum modification. So these are the three most used techniques. Thanks for sharing this information. Elise, your turn.
Yeah, can you go into the next slide? Yeah, thank you. So thank you for this kind of inputs. And now we are focusing more deeply on the chemical penetration enhancer. So ideally, they should be pharmaceutical inert, non-irritant, non-allergenic, non-toxic, cosmetically acceptable, rapid-acting, with predictable duration, reversible, and chemically stable. Chemical penetration enhancers are able to use different mechanisms of action, and it is difficult to assign a primary mode of action for many chemical enhancers. Therefore, they are classified according to their chemical structure. Alcohol are included in the formulation to enhance the solubility of the drug. For example, ethanol will rapidly evaporate at skin temperature. Hamid's family with azone, for example, which was the first compound to be specially designed as a skin penetration enhancer. Esters, and mainly fatty acid esters, included isopropyl myristate, propylene glycol monolaurate like luroglycol FCC, and propylene glycol caprylate with capriol PGMC. Ether alcohol with transcutol are monoethyl of diethyl glycol, which is widely used and reported to increase the solubility of drug in the skin. We have fatty acid-based excipients, and lauric acid and oleic acid are some examples. Generally, unsaturated fatty acids possessing the cis configuration are more effective enhancers. Furthermore, alkene chain length has also an impact. C10 and C12 carbon chain lengths providing reduced permeation enhancement. We have glycol family with propylene glycol as the most commonly used glycol in topical and transdermal product. The pyrrolidone with N-methyl-2-pyrrolidone and at low concentration have been shown to primarily interact with the keratinocyte region of the horny layer, and at higher levels, they also interact with the polar head groups of the lipid bilayer. Sulfoxide, like DMSO, surfactants, terpenes, and of course, the most ideal penetration enhancer with water. So the lipid protein partitioning theory assumes that chemical penetration enhancer can act by one or more of the three mechanisms: disruption of the intercellular lipid domains, interaction with the intracellular protein domains, or increasing the partitioning on the drug into the skin. In the intercellular lipid phase, enhancer may break down lipid packing and or fluidize the lipid chains, creating a more permeable domain. This can be achieved by interacting at three different regions associated with the lipid bilayer. For hydrophilic chemical penetration enhancer, they can interact with the polar head groups, altering the hydration spheres and thus disturbing the lipid packing. This interaction results in a more fluid lipids domain and thus enhance the flux of hydrolipophilic penetrants. Solvents like ethanol or DMSO may act by extracting the lipid from the bilayer, creating holes in the lipid phase. Interaction at the aqueous domain of the lipid bilayer may provoke swelling, increasing the domain volume and changing the bilayer thickness. Lipophilic CPA, on the other hand, are able to insert between the hydrophobic chains, disturbing the lipid packing and thus increasing the lipid fluidity. In the intracellular protein domains, CPA interacts with the keratines, changing its conformation and opening water canals in the cell, facilitating the transport of hydrophilic molecules. And at least, CPAs can be combined with a co-enhancer, water for example, or a mixture of both, to change the stratum corneum properties, increasing partitioning on the drug into the skin. So we will focusing on three different kinds of excipients, starting with transcutal P, which is a purified diethylene glycol monoethyl ether. Owing to a lower dielectronic constant, transcutal P is much less polar than water. Therefore, ionizable drug will dissociate to a lower extent in transcutal P than in water, which favors increased permeation. Another key feature on this solvent is its solubilizing capacity for both lipophilic and hydrophilic API. So this table gives saturation solubility data of a wide set of APIs using topical drug delivery, increasing the drug solubility in the vehicles and thereby the thermodynamic driving force provides higher diffusion and better skin permeation. With no effect on lamellar structure, transcutal P appears to be as gentle as water on the skin. Now going with labrazole, which is a mixture of caprylocaproyl polyoxide glycerides. The main fatty acids are caprylic C8 and capric C10 acids. So it seems that labrazole induces a modulation of the epidermal tight junctions that also may take part in the skin barrier function. The effect of labrazole were compared to sodium caprate, which is a C10 fatty acid known to enhance the transport of drug through the epithelial culture monolayers. Both excipients were applied to emerge reconstructed human epidermis. During the systemic application, the decrease in the transepithelial electric resistance, TEER, was less abrupt with labrazole when compared with C10, even though both excipients produced comparable final effect over time. The effect of this was done on the tissue morphology. Oh, sorry. The effect on the tissue morphology were also observed with light and electron microscopy. So the structure of the only layer, as you can see, was clearly modified after C10 treatment, showing an apparent disorganization in the middle part, whereas exposure to labrazole did not lead to similar deleterious effect. Now, with plural oleic, which is a polyglycerol 3-deoleate, we have done another study with different kind of technique. Starting with a spectrophosphotometry method, investigate whether the skin lipid is disturbed by observing the variation of stretching vibration of CH2, the main functional groups in the ceramide after addition of enhancer. The results highlights a difference in the band value between the control group and when plural oleic CC497 is added, which indicates that plural oleic has a disturbing effect on stratum corneum lipid. As complementary method, a Raman spectroscopy was also conducted, and spectrons were obtained in a region where the difference in lipid can be clearly observed. The smaller the ratio between asymmetric and symmetric stretching vibrations, the greater the lipid fluidity. Addition of plural oleic induces a significant reduction from 208 to 175, indicates that plural oleic was obvious disturbing effect on intracellular lipids domain in stratum corneum. Finally, a confocal laser scanning microscopy is performed to visualize the permeable effect of enhancer in skin barrier. The fluorescent probe Nile fluid was used in this study. For the control groups, Nile red, as you can see on the picture, was detected at 12 micron index, whereas it penetrates up to 24 micron when applied plural oleic. So ex vivo skin permeation tests show that plural oleic has a good effect of promoting drug permeation.
So time for another poll question for our audience. I'm going to share the question. Among Gattefossé penetration enhancers, which one are you using? So capriol PGMC, labrazole, loroglycol FCC, plural oleic CC497, or transcutol P? Of course, you can have several answers. I'll let you a few seconds to answer this poll question. So I will close this poll question and show you the results. So obviously, transcutol is largely used, so which is good news for us, and also labrazole. So which are the two excipients Elise talked about. So thank you for sharing this information. So now, Bruna, we have seen different strategies used for transdermal delivery with a strong focus on chemical penetrations enhancers and how different their mode of action can be. But the question is now, how can we assess the effectiveness of a transdermal formulation? How should we use diffusion cell? And what are in vitro release tests and in vitro permeation tests?
Thank you, Cecile. So before my colleague, Dr. Ashwin Patel, gives you more detailed information about IVRT and IVPT and the differences between those, I would just like to take this opportunity to briefly introduce our experience in the vertical diffusion cell systems field. So in case this is the first time you hear about Hanson or maybe our experience in this field, I would like to let you know that we've been doing these systems for a long time. So it's been about 40 years. We are actually the pioneers in automated diffusion systems. So the microarray technology, which hopefully some of you work or used to work, was the first fully automated diffusion system in the market. And our patented vertical diffusion cell really represented an advancement of the practice many years ago when we launched those systems in terms of accuracy and reproducibility. Then after years working with our customers, and we have customers not only in the industry, but like researchers in universities and regulatory agencies, in 2018, we've led innovation again, and we've launched the system Phoenix, which is a new and improved design of diffusion cell systems. Next, please. Okay.
So next is a question.
Yeah, we have another question. Before we move on to the next slide, one thing that will be I think if you are in this webinar, you probably have some experience with diffusion cells. But we would like to know from you, what's your experience with those instruments?
So still a few seconds to answer this question. So I will close this poll question and show you the results.
Okay, currently working. There's a lot of people that have no experience. Interesting. So we're here to learn. We're glad to be of help. That's great. Okay, so there are two types of diffusion cell tests. So as it was mentioned, we have the IVRT, which is the in vitro release test, and we have the IVPT, which is the in vitro permeation test. And this will be discussed in more details with my colleague, Dr. Ashwin. So we'll just like to start, especially for those of you that don't have experience with the diffusion cells, with the design of the cells itself. So most of the cells, or all of the cells, they will have a donor chamber, which is where you will apply your product, or normally a semi-solid dosage form. And then you have a receptor chamber, which is where you have your receptor solution. In the middle of the donor and the receptor chambers, you have a membrane. That membrane can be a synthetic membrane or a biological membrane, depending on the test that you're doing. And then you have, as I said, you have a product on the top, and then you have the diffusion of the API going through that receptor solution. Then you have a mixer to keep all the cell contents homogeneous here. And you have a sampling arm where you're going to take your samples from, and also do your media replacement. So we do use the same instrument, the same type of test, the same type of cells, sorry. And both tests will involve the detection of an API in the receptor solution at various time points. But those two approaches have different goals and different techniques, and that's what's going to be discussed. One thing I would like to tell all of you, and I'm sure if you're in this field, you've been also experienced that, is that the requirements for those tests are growing very fast. So in the last few years, I would say, especially in the last 3 to 5 years, So I've been working and selling those instruments for about more than 10 years now. And we've never seen so much movement in this area. So people coming from, as I said, the industry and regulatory agencies and universities, researchers, everyone is kind of coming together and putting a lot of attention on these tests. And that is, of course, changing the way that the pharmaceutical and the personal care industries as well perform their tests in their topical and their transdermal products. And that's great because that means better and safer products for everyone. So now I think Ashwin will discuss in more details the differences between Ibrt and Ivpg.
Hello, good morning, everybody. And thank you for joining this webinar. So I'm going to talk about Ibrt, Ivpt, and some of the aspects of those both tests. The requirement for Ibrt is different than Ivpt. So Ibrt is the in vitro release test, which is the assessment of the quality of the product, and it requires to characterize the release rate, and it assesses the rate at which an API is releasing from the drug formulation matrix into the receptor solutions. So Ibrt is established for semi-solid dosage forms like emulsion, cream, ointment, gel, lotions, and all other that type of products. And it has proven to be a successful technique to facilitate formulation development and approval process from the regulatory agencies. And this test is not designed or intended to be correlate with in vivo data. So this test is not going to give any indication of in vivo data comparison. For that purpose, Ivpt test is needed. Next slide, please. So in vitro release test is typically between 4 to 6 hours. Next slide, please. Yes. In vitro release test is typically lasts between 4 to 6 hours. Sampling schedule is typically every hour, but when it's 4 hours or like that, then initially 30 minutes and then increase to 1 hour. But that is also going to depend on when we can see the steady state kinetics. Synthetic membranes are used for in vitro test. It's a non-physiological media like hydroalcoholic as 30% ethanol or something like organic and water mixture is used. Occluded top to prevent drying and metamorphosis of the product on the diffusion cells. Pseudo-infinite dose is used and steady state kinetics are achieved. The primary results is steady state release rate, means the slope at which the API is releasing from the product matrix to the receptor solution. And drug is released into the receptor solution in microgram per milliliter concentration range. Next slide, please. So this is the technique where you can see how the cell preparation is going to happen.
I'm going to demonstrate how to set up a cell for an Ibrt test. So we're going to take our diffusion membrane and place it in the bottom portion of the cell cap. And then we're going to take the top portion of the cell cap and put it on to secure the membrane in place. Next, we're going to take our receptor solution and fill the cell. We'll put our cap into place and we'll tilt our cell to fill the rest of the way. The cell is fully filled with media when the meniscus passes through the center of the white dot on the sampling arm. Once it's assembled, we can put it into its position on the cell block. Now we're going to allow the receptor solution and membrane to equilibrate to the desired temperature. Once it's reached the desired temperature, we're going to take an infrared thermometer to measure the surface temperature of the membrane. Once we see that it's at the temperature, we can begin to prepare the sample. So I'm going to take our sample and apply it into the dosage chamber to the membrane. We will then take our spatula to remove any excess and remove any bubbles from the dosage chamber. Now our sample is fully applied. We will take a clear dosage lid and cover our sample. Now we're able to start the test and repeat the process for all cells.
So as you can see that sample preparation for an IVRT test is really very easy with the techniques shown in this video. Now, let's say if somebody is asking that what will be my receptor solution, because we talked about it, that receptor solution is important and all the stuff. So receptor solutions should not be reactive to the membrane or to the product. It must achieve the diffusional sink condition, and it should increase the solubility of API, typically being able to hold 10 times of the total amount released from this IVRT study. Commonly used solutions as a receptor solution is hydroalcoholic media, which is 30% ethanol, 30% isopropanol, or 1-butanol, or even with a higher percentage of those is used. DMSO and THF, so dimethyl sulfoxide and tetrahydrofuran, can be used at various percentages with the water or with other miscible solvent. And when the other solvents are not used, these are like a more aggressive solvent, but it can be used. And also use of surfactant is acceptable, but there should be a justification that why we are using. Normally all other receptor solutions are not providing desirable result. That's how we are using the surfactant. And evaluation is done using a minimum of 3 receptor solutions in each in triplicate diffusion cells, and also in a 3 times study with the API into the receptor solution to find out the best solubility and the best receptor solution. Next slide, please. Yeah, now choosing of a membrane, because the receptor solution and the membrane are the most critical parameters or most critical item to choose for IVRT studies. So that depends on API and the product matrix. Synthetic membrane, mostly 0.45 micron and 25 millimeter size, commonly used, but there are many other membrane sizes available in terms of its porosity. So if we have hydrophilic API, we should choose hydrophobic membrane, and if we are working with hydrophobic API, we should use hydrophilic membrane. So what will happen with this, that it is reducing and eliminating an issue of API binding to the membrane, because API binding to the membrane is one of the study we have to prove into the method validation. Pore size of the membrane should be large enough that the API will easily diffuse through, but not the other component from the matrix. We should try to eliminate that. So nylon, PES, acetate, mixed cellulose, or PTFE are the most commonly used synthetic membranes. And the more important thing is also the consistent supply. Evaluate a minimum of 3 membranes, each in triplicate, with triplicate diffusion cells in triplicate studies to evaluate API bindings with the membrane. So membrane suitability is the one which we are trying to find in these triplicate studies. Next slide, please. So now we are talking about IVPT. As I said, that IVRT and IVPT are two different tests. So IVPT is used to understand the behavior of the components that mimic the travel through the skin. It means permeation. So how the product is going to permeate when it's applied in a real life scenario. So vital to guide the product development and fundamental is the understanding efficacy of the formulations. So IVPT test is going to correlate with your in vivo data, and that is expected in this study. And it is the assessment of the product performance and safety. So in vitro permeation test, which is little longer, not little, it's more longer than the IVRT studies, and typically 24 to 72 hours. And biological membrane, which is like skin, as Bruna said before, that biological membrane are used for IVPT studies. Physiological media are preferred for this type of studies and often not included, means exposed to the air when the sample is applied. So as I said, in IVRT, we are going to close the cells from the top using a glass or a rigid material to close exposure to the air. But here we have to keep it open, and if there is any drying or metamorphosis is occurring, we have to let it happen and then do the studies. So it's a more often it's a semi-finite dose for a non-steady state kinetics, and the primary results is dynamic rate of the permeation, means we are going to obtain the flux profile in these studies, and drug is often released into the receptor solution in a nanogram per milliliter range. So if you remember in IVRT, I said that it would be in milligram per milliliter range. So here is much lower. Next slide, please. Now it's the same thing for IVRT and IVPT. Receptor solution is very important. So normally in IVPT test is a phosphate buffer saline, is known as PBS, with or without additives like antibacterial, typically gentamicin is used. pH to be adjusted in the range of 5 to 7 to reflect the physiology of skin. Viscosity should be low enough to enhance permeability. There are some other factors, as discussed previously, that how permeability can be increased, but we should try to have low viscosity receptor solution. Must be non-reactive with skin and the product which we are going to do the test on, and it must maintain the sink condition. Next slide, please. So IVPT, the membrane. Normally the cadaver skin, surgical skin or animal skins are used. Anatomical location like posterior torso or abdominal part skin is used. Permeability is inversely proportional to the skin thickness. If the skin sample is thick, then permeability will be lower. So right size of the skin is very important in this study. Skin preparation is very important, as dermatomed or a heat separated epidermis, etc. are used for the study. The barrier integrity test, normally, like most commonly people do this test, is a trans-epidermal water loss test, which is TEWL, and use the vapometer to do this test. And again, here also the consistent supply is very important, because skin has its own variability where it is coming from and which body part is used for the study. So consistent supply is very important. Next slide, please. So now it's about the regulations and guidelines. Even though relatively this is a new studies, new test, however, it's being done since many years, but there are many regulations and guidelines are published by various regulatory bodies, and Bruna is going to talk about that. Thank you.
Thanks, Ashwin. So there is a number, as Ashwin said, of guidelines on those tests. So I would say the mostly used by our customers, we have the European Medicines Agency guidance, the draft guideline on quality and equivalence of topical products. We have the OECD 428, which is the skin absorption in vitro method, USP chapter 1724 for semi-solid performance tests, FDA draft guidance on acyclovir. We have, of course, from FDA the SUPAC. There are other documents as well. But when you go through all of those documents, there are a few key points that are kind of repeated in all of them. And I would like to share those with you, which is key for success in IVRT and IVPT testing. So the first one, as it was mentioned before, the diffusion test by itself, especially IVPT test, it has its own variability by itself, especially when you're working with real skin. So the first one is to minimize variability as you can, as much as you can. So either in the instrument or in the skin that you're working, the procedure that you're doing, so minimize variability. Second one, good temperature and mixing control. Those are probably the two key aspects. We will discuss that later in the presentation again, in the instrument itself. So have a good temperature and mixing control. Method robustness, we have the manufacturing consistency, not only the instrument, but it was mentioned before about the membranes as well. Easy to prepare and to sample, so a system that is easy to set up, to clean after, to sample, and so on. Well-controlled study procedures and precision and reproducibility. So those are the key points for a successful IVRT and IVPT. Next.
So thanks for explaining how to perform in vitro release tests and in vitro permeation tests, how to choose a membrane and receptor solutions. It was very interesting. But let's see now how a complete formulation should be developed and assessed on diffusion cells.
Thanks, Cécile. So now going deeply on the solubility and permeability. As you probably know, the rate of the drug transport across the stratum corneum follows a Fick's law of diffusion, where the drug is applied to the membrane at a maximum fixed concentration in the donor compartment and under skin conditions in the receptor compartment. A steady-state Fick's first law can be simplified as GS equal to dKm CV divided by H, where GS is a flux at steady state, D the diffusion coefficient, K is a vehicle membrane partition, C is a concentration of the drug in the vehicles, and H the length of diffusion pathway or the skin thickness. With this parameter in mind, effective permeation enhancer can increase transdermal drug delivery by a change in D, K, and C. Therefore, compounds which are skin penetration enhancer may potentially change the solubility partitioning behaviors of the drug into the stratum corneum and or its diffusion properties. So skin delivery of any active ingredient can be controlled by the choice of the polarity of the phase in which the active ingredient is incorporated. So there is two important things to respect. On the one hand, there should be enough drug in the formulation to allow its minimal effective concentration to be reached at the target site. This is achieved by using a primary solvent in which the active ingredient is very soluble, such as the highest absolute solubility is achieved. On the other hand, a formulation containing only this primary solvent will not deliver the active ingredient very well because its driving force for diffusion is far from optimized. So the active ingredient simply likes the formulation too much. This driving force to leave the formulation can be achieved by adding a secondary solvent which the active ingredient is not very soluble and that pushes the active ingredient out of the formulation into the stratum corneum. So this creates a low relative solubility on the active ingredient relative to that in the stratum corneum. So in other words, the solubility of the drug in the stratum corneum should be higher than that in the formulation, but the solubility in the formulation should be high enough to allow a sufficient drug to be dissolved to be able to achieve a clinical efficacy. So some excipients could act increasing the drug solubility in the stratum corneum, like transcutal P, and in combination with excipients that increase the diffusion of the drug in the stratum corneum, like loroglycol FCC or capriol PGMC, this can lead to a synergic effect, what we call the push and pull effect. So Gattefossé offers a large range of solubilizer for topical delivery system. They are all liquid, non-toxic, non-irritant, conform to pharmacopoeia, and already approved. So they display solubility properties or help the drug to partition and diffuse through the different layers on the skin. So we have the propylene glycol esters with the capriols, loroglycols family and Labrafac-PG, the PEG esters with the Labrafil and Labrazole, Labrafac lipophil and PCol for more lipophilic molecules, Furololic CC497, which is a polyglycerol esters, and we have already focused on it, and of course the transcutal P. So going now to some case study, starting with clonazepam, which is a long-acting benzodiazepine commonly used to treat panic disorder and severe anxiety. So these examples highlight transcutal P behavior and the importance of using a mixture of solvents, keeping in mind that water is key solvent in topical formulations. So in this study, 200 milligrams of gel formulation were applied on rabbit hair skin, and the permeation of the drug was assessed in vitro. So with rising transcutal P concentration, both the flux and the solubility of clonazepam increase, and at 50% of transcutal P concentration, the right balance between solubility and permeation is achieved. Now, second example coming from our lab with lidocaine hydrochloride. So its solubility was assessed in various solvents and penetration enhancer, either alone or in combination. In neat excipients, highest solubility was obtained for water, followed by transcutal P, whereas loroglycol FCC, propylene glycol monolaurate, exhibit a limited solubility. Permeation of lidocaine hydrochloride was assessed in Franz cells using pink skin for transcutal and loroglycol or capriol PGMC alone or in combination. In neat excipients, highest permeation rate was obtained with capriol PGMC, followed by water and transcutal P. So this shows that also water is the best solubilizer for lidocaine hydrochloride. It is not the best permeation enhancer. So when combining loroglycol FCC or capriol PGMC and transcutal P, a faster release of drug diffusion was observed, demonstrating a synergic effect. So transcutal P increased the drug solubility in the formulation, whereas loroglycol or capriol push the drug to partition into the membrane. So next slide, please. Yeah.
So time for another question. What type of dosage forms are you using? So creams, gels, ointments, solutions, patches? I'll let you a few seconds to answer this question. So I will close this poll question very shortly. And show you the results. So creams and gels come first. Thanks for your answer.
Thank you. Thank you, Cécile. So going more deeply on the protocol now, as you might know, careful consideration should be given to the method of preparation of the skin to be used in the in vitro experiment, as both permeability and lag time can be greatly affected. The dermatoned skin is common preparation technique requires some skill on the operator to achieve a consistent result. So OECD, WHO, and FDA guidelines recommend the evaluation of skin integrity prior to the permeation study, as Ashwin also mentioned in the previous slide. So this can be achieved by either measuring the permeation of marker molecules like titrate water, caffeine, or sucrose, or by physical methods such as the determination of trans-epidermal water loss or transcutaneous electrical resistance. So now, to illustrate how we can prepare the skin and how it's evaluated, a video has been done in our laboratory, and we are very pleased to share it with you today.
The pig skin is stored in the freezer at -20°C. Before being used, it is unfrozen by dipping in water at ambient temperature for 5 minutes. A dermatome is used to collect the upper 0.8 mm of the skin. The skin is visually examined for its physical integrity. If there is any injury or tattoo, this part of the skin is discarded. A series of skin discs are punched in the skin. Their thickness is evaluated into 2 glass plates in 5 points. Note that it is important to have full traceability of each skin sample, and each disc is duly attributed to a cell number. When the skin disc is ready, it is set on the Franz cell, and carefully applied to ensure proper sealing. Then the top of the cell is set and clipped. When all Franz cells are equipped with the skin discs, the receptor fluid is allowed to circulate in the cells for 15 minutes. This step enables to check the quality of the sealing and the skin integrity. Cells are gently upturned to remove any air bubbles. Trans-epidermal water loss is assessed on each cell. Measures are repeated 3 times on each cell, and the results carefully reported. The formulation is applied on the skin. It is important to control the exact quantity of formulation that is deposited on the skin. A cap is added to close the system. Finally, the programmed protocol is launched, and samples will be retrieved at regular intervals for 24 hours.
So as you see on this video, we use the Ibrt Hanson Franz cells, which is not the new one, not the Phoenix, but the protocol it's the same. Okay, so going to the storage condition now. While it is sometimes possible to transport tissue directly from the operating theater to the diffusion cells without freezing, under most circumstances, the skin will be frozen prior to processing. Also, some authors conclude that freezing has no measurable effect on permeability. It is therefore important to check and prove it by conducting internal study. And moreover, there is no consensus of the storage duration. For example, the International Program of Chemical Safety states that human skin can be stored to 1 year, whereas the US Environment Protection Agency allows the storage for up to 3 months. So in our lab, we have evaluated the performance of the same formulation, it was lidocaine hydrochloride-based gel, for 7 months. This test has been done on pig skin with the same donor, with a minimum of 6 replicates for each test. The graph provides the percentage of lidocaine hydrochloride that penetrate the skin for 24 hours, and its kinetics over time. So T0, 1 month, 3, 4, 5, 6, and 7 months. So except for the 4 months point, the results suggest that the performance is still similar during this period of time. Same kinetics, same lifetime. So after a deeper analysis, the poor performance of the 4 months test was due to the pink skin area used. So it could be concluded that performance of the formulation can be conducted for 6 months since the permeability of the pink skin is not affected. So as Ashwin and I think Bruna already said, it is really important to check the robustness on your result and protocol. So the consistency in tissue retrievals, handling, storage can minimize erroneous data, and accurate tissue information can help determine if the results are valid. In addition, all data should be included in a statistical analysis. In our study, so the same formulation was evaluated on the same donor. We have plotted all the variables we have generated: pink skin age, application area, amount of formulation applied to the membrane, TIWL, and the skin thickness. So the diffusion at 24 hours was the selected response in this example. So just the key information, the protocol that we have used are carefully be designed to minimize the data variation, like for example, the quantity applied. So as you can see from the different graphs, points are not gathered around the line, suggesting that the age, the quantity of formulation, the TIWL, the skin thickness has no impact on the diffusion. And yes, however, when looking at the statistical analysis for the zone of application, the data highlights its impact. So it seems that the extreme area, so close to the pink's head and its tail, display different permeation characteristics. This zone should be hence be excluded from the study. So our results suggest that careful standardization and validation of the protocol for in vitro skin studies should be carried out prior to the experiments for obtaining reliable data.
Okay, I'm back. So now we're going to discuss the key aspects of the instruments. So I would say the most important aspects would be temperature control and mixing control, as I've mentioned before, or the stirring here in this presentation. In terms of temperature, customers will work most cases at 32 degrees. We do have a tolerance in the guidelines that says plus or minus 1 degree C. That's the tolerance. There are 2 ways of controlling the temperature. So we can do it through a water bath circulator, and normally we have water jacket cells, and that will keep all of the cells at temperature. Or newest systems, where this is our Phoenix DB6 system, we work with a dry heat block. So this block will heat and mix all of the cells contents at the same time. And what this offers the better temperature control. You have a very fast heat up compared to the circulating bath. And of course, you don't have all of those tubing that will require some cleaning and maintenance. Then we have the mixing or the stirring. So again, we do have a tolerance here. Normally, customers will work between 400 RPM to 600 RPM. Of course, it can be different, but that's commonly used. And we do have a tolerance of 10%. That's the tolerance for the speed here for the mixing. There are again 2 ways of doing. So commonly used is those magnetic stirring bars. But then again, with the Phoenix systems, we improved and we redesigned those mixers, and we came out with those grooved mixers. So those mixers here will offer a better homogenization and will prevent any unmixed or high concentration API under the membrane, which will slow down the diffusion process. And we also redesigned that arm here. So if you are familiarized with the model B and C in the USP 1724, which you have a very thin and long arm, we now have a shorter and larger arm. And this, together with that mixer here, which is a more robust way of doing the homogenization of the cell, it will ensure that you have concentration uniformity in the body of the cell and in the sampling arm as well, because that was another source of variability, of having one concentration in the body and a different concentration in the arm of the cell. So here we have a performance study that we did that proves that within 5 seconds of mixing, we have the same concentration in the body and in the arm of the cell. So we're reducing or minimizing variability, again, which I mentioned is very important for this test. Next, please. Okay, then the volume of the receptor solution. So it was discussed the types of receptor solution and that you must meet and maintain sink conditions during your test, and that's very important. So to address that issue, what we've done, we have 3 sizes of cell. So we have a small, a medium, and a large. And then the mixers, we have 2 types of mixers, a tall mixer and a short mixer for each one of those cells. And that will allow for adjustment of the volume on the cells, helping our customers to ensure sink conditions. So when you're working with normally a set of 6 cells, so the nominal volume for all of the 6 cells should be the same. So if we're working with a small volume cell, all of them should be, or a 10 ml cell, all of them should be 10 ml. But then true volume should be measured for each individual cell. That's very important as well. Then we have the cell orifice diameter. That's the membrane exposure area. So here's basically where you will apply your product, and then it will be in contact with your receptor solution and your product. So the amount of API entering the receptor solution here is directly related to the exposed surface area of the membrane, which will depend on this orifice diameter. So again, there is a tolerance here as well. So the tolerance between the orifice of the donor chamber and the receptor chamber, it should be within plus or minus 5% of the specified diameter. In the Phoenix system, we machine those parts into very, very tight tolerances. So we're talking about 0.01 millimeters. So that meets and exceeds actually the tolerance that is in the guidelines. So again, another source of variability that is minimized here. Next, please. Okay, then we have the sampling and media replace volume qualification. So here, because we are working with very small volumes, so I don't know if you could see in the previous slide, but we're talking about in the Phoenix cells from 10 ml to 30 ml volume, and there are cells with even smaller volumes. So it is very important that every time that you withdraw the sample, you have to replace that media. Not like this solution where you work with larger volumes. Some methods will have media replacement, some will not. Here you do not have an option. Every time you take your sample out, you replace with new media. So if I'm taking 200 microliters, let's say, I'm replacing with 200 microliters. So there are 2 ways of sampling and doing your media replace. Of course, you can do it manually. So here it's showing our Phoenix DP6 system. You have to find a system that is easy to sample and that is precise and reproducible. Here, as you can see in the Phoenix, you can do, because of, again, that large arm that we've designed, you can sample with a regular pipette. You don't need those type of capillary accessories that sometimes you need to sample from other cells. So that makes easier and more reproducible, I would say. or you can do through an automated, this is our Phoenix robotic diffusion station. So the volumetric accuracy the system works with a precision syringe, which is extremely accurate, of course. So here we're talking about the specification of the system, 0.025 mil is the stated specification. So again, it's a very accurate way of doing your sampling and your media replacement automatically. Next, please. Thank you. Here I wouldn't say are must-have features, but nice to have. So a system that is easy to set up and that offers a flexible design. So for previous systems or other systems, they have clamps and they have different parts that you have to put it together to mount the cell. So one thing that we've done are those cell caps that they align together and they click together very easily. I think you could get an idea from the first video, the IVRT video, on how easy it is to set up. Then the dosage form are applied to an easily accessible opening in the cell cap, and the flexibility. As I mentioned, we can accommodate different volumes on the cell for zinc. We can do different dose volumes as well, different types of dosage volumes that we can accommodate. If it's more viscous, like semi-solid, or less viscous if it's a solution, or if it's a transdermal patch. Orifice diameters, as I mentioned, is the exposure membrane area. We can also adapt that. The type of membrane, if it's a synthetic, a biologic, maybe it's a full thickness skin. So we can accommodate all of this flexibility, different solvents as well that customers may use, more aggressive, as Ashwin mentioned. Also important, all components are serialized for easy traceability. So the body of the cell is serialized, the mixers, the cell caps here. So like in dissolution, where we normally recommend that you keep the vessel and the apparatus all in the same position and kind of group them together and always use them together. Here is the same situation. We recommend for easy traceability that you always keep the same cell with the same mixer with the same cell caps. And that's much easier to achieve if all the parts are serialized. Next, please. Well-controlled study procedures. That's also mentioned in some of the guidelines. So in both our manual system and automated system, we give our customers the capability to have full control over their study procedures. So it's not like you can only change, mostly manual systems, you can only change temperature and speed. Here you can have all the information. This is our manual system. The customer can include all information about their methods. You have a complete audit trail. You have up to 50 users can be password protected with different privileges. So that also helps with 21 CFR Part 11 compliance. And then you have this enhanced with our Phoenix robotic diffusion station, which is controlled by a 21 CFR Part 11 diffusion master software. And here, of course, you will put all the information about your method, about your sampling time, sampling volume, rings, media replacement, and the system will do everything automatically for you, which makes life, especially for IVPT tests, where again, Ashwin mentioned you can have tests from 24 hours up to 72 hours. And we know customers that have even longer tests, 96 hours, for example. So it makes life easier if you have an automated system, of course. You also have all the reports saved and can be approved in the software here. And in terms of reports as well, it's very complete. So you do have better control over your study procedures. Next, please. Okay. This is just a quick tip on air bubbles detection. So if you've been working, and most people here have been working or worked in the past with those cells, or if you're new to diffusion cells, bubbles are quite of a big issue, I would say. So this is another source of variability. And if you have a bubble between the membrane and your receptor solution, then the diffusion won't happen. And that's an issue. So instead of you kind of going under the membrane, trying to look what's going on, if there is a bubble, if there's not a bubble, we sell with all of our instruments this lighted mirror here, but that's very easy to find on Amazon or some tool shop. And it's a mirror, as you can see here, you can easily detect the bubbles on a more ergonomic position. So I think your spine will thank me for that one. And if you find a bubble, you can just easily tilt a little bit here the cell and get the bubble removed here in the sampling arm. I think that was also shown in the video. So air bubbles detection, very easily done. Another tip about transferring methods. So we do get asked a lot about customers either using the microet or other systems, and they want to start using the Phoenix, and they are concerned about the method transfer. So everything that we've discussed before will have, of course, to be evaluated. So we talk about the volume of the cell and the receptor solution, the mixing, the orifice diameter, the sampling volume and media replacement volume, the procedures for dosing the cells. So some cells you dose before, some cells you dose after it came to equilibration, the chemical compatibility of the materials on the one cell and the other. So running comparison tests between existing systems and new systems is always recommended. I mean, we've mentioned many times that diffusion testing by itself has a lot of variability. So the evaluation should use a set of data that is statistically significant. So in most cases, 6 tests of 6 cells in each system should be sufficient to have that data, to have statistically significant data for your method transfer. One thing, if you're thinking about transferring a method, please do reach out to me or to Ashwin, or either in our website, LinkedIn, any of our channels here, that we can definitely help you with the method transfer from one cell to the other. And Ashwin, I don't know if you want to add anything to this slide, any comments in terms of method transfer?
Thank you, Bruna. You said it correctly that yes, we can definitely help whenever it's needed, but if we consider that what is the most important parameter to think about, it is orifice diameter and the volume in the receptor solution. That could be the major variable here, but orifice diameter is going to remain same because our design did not change. So whatever was available on microet system is available on diffusion Phoenix platform. And the volume, yes, it is different, but that volume can be studied and optimized by injection volume on HPLC method, because this will be little more diluted into receptor solution, but you can have larger volume of injection into the HPLC method. And the sampling volume, as typically I have seen people were using 400 microliter or something, which is also possible in our system. So we try to mimic as much as possible parameters to remain close with the microet system. And definitely we can reach out to us if it's needed. Thank you.
Thank you.
Okay, so thank you to all of you. That's the end of the presentation. Thank you to the speakers. It was a lot of information. And let's now start the Q&A session. So just trying to increase the size of the window to be able to read the question. Sorry about that. A question for Elise to start with. Which chemical penetration enhancer mechanism of action is the most damaging to the skin?
So normally a chemical penetration enhancer should be inert and have no effect on the skin. But as I said in a previous slide, there is some chemical penetration like DMSO that creates some holes in the skin, which is very efficacious because it will penetrate through the skin, but it will also damage the skin. So this kind of excipients, chemical penetration enhancer, that are more known to be irritant and create some holes in the skin are now to be excluded from the study because they are quite irritant.
Okay, another question. Well, sorry, still for you, Elise. How do you get the diffusion coefficient you have cited in your equation?
The coefficient, can you repeat, please? The coefficient?
How do you get the diffusion coefficient you are using in your equation you showed?
Yeah, it could be done by the partitioning between the water and octanol. It's like Loki or something like that. You can calculate it easily.
Okay, thanks. Can strat M membranes perform similar to skin?
Sorry?
If you use the strat M membranes, do they perform similarly as the skin?
I can answer, but Ashwin can complete, I think. We start with strat M. I think just to be trained on Franz cells, because that's the first time that we have this kind of study. It's great to start with, and if you have a look to the different papers, they are claiming that there is a good correlation between strat M and pig or human skin, but there is some constraints by using this synthetic membrane. And if you want to have permeation test, it's really mandatory to use animal skin or human skin to be more relevant, because you cannot have some correlation regarding the structure of the skin, the composition, if you compare human and stratum membrane.
Okay.
Maybe Ashwin will maybe complete this.
No, exactly what you said, please, is correct. Thank you.
Is it essential to saturate the membrane, for example, for strat M? Should I saturate it with the receiver media before use?
Actually, I have seen that the product insert says do not need to do that one. The test can start it immediately without saturation on stratum membrane. That is listed and labeled, and that's what the practice people are doing. But maybe Elise, you can share your experience and opinion on that.
We test with and without saturation on the membrane, and we didn't see any improvement of saturated or not saturated the membrane. So for us, it was not needed to do this step.
Yeah, so that means the product label, because we also use them, and that says that can use immediately without saturation.
Exactly, yeah.
Do the guidelines specify any dimension specifications for the receptor volume, the speed of stirring, the area of the cell, and so on?
Yes, we do. I've mentioned during the presentation, so for the temperature and for the mixing or the speed, you do have for temperature plus or minus 1 degree C, and the tolerance for speed plus or minus 10%. Then you also have for the construction of the cell, let's say, so it's all diameters can have a 0.5 millimeters tolerance, and all lengths 2 millimeters tolerance. And then for the alignment of the receptor orifice and the donor orifice, you can have 5%. It's a 5% tolerance. So there is a specification for all of this, and tolerances that must be followed.
For the EVPT performance, it was mentioned that only the exposed surface area determines the result, but does the total size of the membrane has an impact on the result? Is there distribution inside the membrane?
Do you know the answer, Ashwin? I didn't really understand the question, to be honest.
I shall repeat it if you want. For the EVPT performance, it was mentioned that only the exposed surface area determines the results, but does the total size of the membrane impact the result? Is there distribution inside the membrane?
I can just share what I think about it, because the way how cell design is made, that the membrane is going to remain with the other solid part of the cell structure, and it should not impact. It will, the same thing is going to happen, and because of that reason also the membrane binding study is performed in matter development and validation, which can prove that no other that material is not binded to the membrane, and whatever is into the receptor solution, it's coming from the membrane, and there should not be anything to hold onto the membrane, either it's a contact part or non-contact part of the membrane. Because you know, if you imagine from through the orifice the diffusion is going to occur, and the other part there should not be any diffusion going. So I don't believe that anything is going to be stayed on that membrane which is not open through that orifice. Alice, any opinion, anything you can share?
No, to be honest, I didn't catch the question, because
actually what it is, they're trying to understand that anything is going to go beyond that orifice area onto the membrane, like molecule is going to pass.
Yeah, but I think when you work, they mentioned IVPT, so when you're working with real skin, if you have let's say 25 millimeter skin, I mean, the person who asked me clarify if I understood correctly, if the product can be, and then let's say we have a 13 millimeter orifice, if the product can kind of go through other parts of the membrane. Maybe the person can clarify the question.
Yeah, it could be the way that this person give more information, clarify his question, and we'll come back later on. Because time goes fast, and I still want to have one or two other questions. Can we understand the distribution of the drug that has permeated along the various layers of ex vivo model, when skin, for example, during IVPT studies? Can we have the amount of drug permeated in the epidermis, the dermis?
Yeah, but in this case, you have to do what we call the mass balance, meaning at the end on your kinetics, you remove your skin and you analyze what is on your skin, inside your skin, and go through the skin. So you have some distribution on the active to different layers. And if you want to have deeper information about the different layer on the skin, you have to do, for example, the stripping, meaning that you put some strip on your skin and you put the different layer on the skin and you analyze the different layers, so you have an idea about where your drug is located in the different layer.
Yeah, I think commonly used from customers to take the stratum corneum, you use the tape stripping. Then I don't know, some of the protocols say like 20 tapes will then take out the stratum corneum, and then you have a way, I think you heat the rest of the membrane, and then you can separate the dermis and the epidermis and evaluate them separately. So it is possible.
So time for a last question, because it's already noon. What is the suggested measure for practically water-insoluble drugs?
In the receptor chamber?
I assume it's not specified, but I assume it is the case.
You know the answer, Ashwin?
Yeah.
Typically, this is like a very typical question, and also it should be studied with various hydroalcoholic media. Because if it's a water insoluble, only that organic part is going to help dissolve this material. So based on the solubility of the API in which type of organic solvent is soluble, you can mix and use that one. Alice, your opinion, please.
Yeah, it could be hydroalcoholic, or you can put transcutol into your receptor as well.
Yeah.
Because to be sure to have the sink condition. But yes, you have to evaluate the solubility of your active in the receptor to be sure that you have in the sink condition. So adding some surfactant or some solvent or transcutol could help to achieve this condition.
Okay, so thank you very much to all of you. We are reaching the end of this webinar. It's already 1 hour and a half that we are together. So thank you very much for joining us. There are still a lot of questions, and some people have really concrete projects with many questions. So they will be contacted directly later on to answer these questions. Thank you very much for joining us. The on-demand webinar will be available on our website next week. So if you need to go back to access this information, it will be easily accessible. And also you will have a survey at the end of this webinar. And if you could please take a few minutes to answer it, it will help us improve our next sessions. Thank you very much. Thank you to all of you. Thank you to the speakers as well. And hope to see you very soon.
Thank you.
Thank you so much.
Thank you.
Have a nice day, everybody.
Bye.
Bye.
Bye.